What are free radicals? These mysterious molecules are at the center of many discussions about health and aging. Free radicals are unstable molecules with unpaired electrons, which makes them highly reactive. At their core, free radicals are atoms or molecules with an odd number of electrons. Unlike stable molecules that have paired electrons, free radicals desperately seek to pair with other electrons to become stable. This chemical instability is what makes free radicals potentially harmful to the body. Free radicals often steal electrons from other stable molecules, turning them into free radicals too, creating a destructive chain reaction. This damage, known as oxidative stress, can harm cells, proteins, DNA, and other important structures in the body. Understanding the electron structure of atoms helps us comprehend why certain molecules are more likely to become free radicals. In summary, free radicals are unstable molecules with unpaired electrons. This makes them highly reactive and eager to steal electrons from other molecules. Oxidative stress occurs when free radicals overwhelm the body's natural antioxidant defenses. Free radicals damage cells by stealing electrons from nearby stable molecules. This creates a chain reaction where the newly formed radical steals electrons from other molecules. Lipid peroxidation occurs when free radicals attack the cell membrane. This creates a chain reaction that damages the membrane structure, leading to cell dysfunction. Proteins are also vulnerable to oxidative damage, which changes their structure. When oxidized, proteins unfold, losing their normal function, which can lead to cellular dysfunction. DNA is particularly susceptible to oxidative damage. When DNA bases are oxidized, it can lead to mutations. One example is the formation of 8-hydroxyguanine, which causes GC to TA transversions. The damage caused by free radicals accumulates over time, contributing to aging and various diseases. This is why antioxidant defenses are crucial for protecting our cells against the cumulative damage caused by free radicals. Hydroxyl radicals are highly reactive free radicals that can attack DNA in multiple ways. Free radicals can damage DNA through three main mechanisms: single-strand breaks, double-strand breaks, and base modifications. First, hydroxyl radicals can attack the sugar-phosphate backbone of DNA. This weakens the structure and can result in single or double-strand breaks. Second, free radicals can cause chemical modifications to DNA bases. These include oxidation, deamination, or even complete loss of bases. Cells have evolved sophisticated repair mechanisms to fix DNA damage. However, these systems aren't perfect. If damage isn't repaired correctly, it can lead to permanent mutations in the DNA sequence. DNA repair enzymes identify and correct damage. When working properly, they restore the original DNA sequence. When DNA damage affects genes that control cell growth and division, it can lead to cancer development. As mutations accumulate, normal cells may transform into cancer cells. To summarize, free radicals damage DNA by causing strand breaks and base modifications. While cells have repair mechanisms, some damage may become permanent mutations. When these mutations affect genes controlling cell growth, cancer risk increases. We'll explore how free radicals and inflammation form a complex, bidirectional relationship in the body. The cycle begins with tissue injury, which can be caused by trauma, infection, or exposure to toxins. When tissue is damaged, the body initiates an inflammatory response. Immune cells are recruited to the injury site. These immune cells produce free radicals as part of their defense mechanism to destroy pathogens. The free radicals are highly reactive molecules with unpaired electrons. However, when produced in excess, free radicals can damage healthy tissue cells. This happens when the free radicals react with cell components, oxidizing them. Free radicals can directly activate inflammatory signaling pathways within cells. When a free radical interacts with a cell receptor, it triggers a cascade of signals. This signaling pathway activates genes that produce inflammatory mediators called cytokines. Cytokines are released from the cell and amplify the inflammatory response. This creates a positive feedback loop. Tissue damage triggers inflammation, which produces free radicals. These free radicals cause oxidative damage, which leads to more tissue damage, perpetuating the cycle. When this cycle continues unresolved, it leads to chronic inflammatory conditions. In rheumatoid arthritis, persistent inflammation and free radical damage occur in the joints, causing pain and tissue destruction. In summary, free radicals and inflammation form a bidirectional relationship. Free radicals can trigger inflammatory responses, while inflammation produces more free radicals. Breaking this cycle is crucial for preventing chronic inflammatory diseases. The free radical theory of aging proposes that cellular damage accumulates over time due to free radical attacks. Free radicals cause oxidative stress, damaging cellular components like DNA, proteins, and lipids. Free radical damage to DNA is linked to various age-related diseases through complex cellular pathways. In cardiovascular disease, free radicals oxidize LDL cholesterol. This oxidized LDL contributes to arterial plaque formation. Neurodegenerative diseases like Alzheimer's and Parkinson's are linked to oxidative stress in brain tissue. The brain is particularly vulnerable due to its high oxygen consumption and limited antioxidant capacity. In type 2 diabetes, free radicals damage pancreatic beta cells that produce insulin. Hyperglycemia and hyperlipidemia increase ROS generation, causing oxidative stress that impairs insulin secretion. In cataracts, free radicals cause protein oxidation in the eye lens. This oxidation leads to protein cross-linking and aggregation, causing the lens to become cloudy and vision to blur. Free radicals cause comprehensive damage to cellular components, including lipids, proteins, and DNA. This damage accumulates over time, contributing to aging and the development of age-related diseases. Antioxidants can help neutralize free radicals, potentially slowing the aging process and reducing disease risk. The human body has sophisticated defense systems to combat free radical damage. The body employs two main types of antioxidant systems to neutralize harmful free radicals. These include enzymatic antioxidants and non-enzymatic antioxidants. Enzymatic antioxidants are specialized proteins that catalyze reactions to neutralize specific free radicals. The main enzymatic antioxidants include superoxide dismutase, catalase, and glutathione peroxidase. This diagram shows how these enzymes work together in the peroxidation process. Superoxide dismutase converts superoxide radicals into hydrogen peroxide, which is then neutralized by catalase and glutathione peroxidase. Superoxide dismutase, or SOD, is our first line of defense. It converts the highly reactive superoxide radical into hydrogen peroxide. Catalase and glutathione peroxidase then convert hydrogen peroxide into harmless water and oxygen, preventing further cellular damage. Non-enzymatic antioxidants are molecules that can donate electrons to free radicals without becoming reactive themselves. These include molecules like glutathione, vitamin C, vitamin E, and various carotenoids. This diagram illustrates how chain-breaking antioxidants prevent radical chain reactions, particularly in lipid peroxidation. These antioxidants can be donor types that transfer hydrogen atoms or sacrificial types that form stable products with radicals. Let's examine how antioxidants neutralize free radicals through electron donation. Free radicals are highly unstable because they lack an electron. Antioxidants can safely donate electrons to neutralize these radicals. The free radical becomes stable after receiving an electron, and the antioxidant remains relatively stable even after donating it. This mechanism effectively breaks the chain reaction of free radical damage. Instead of attacking cellular structures, free radicals are neutralized before they can cause harm. A diet rich in antioxidants is one of the most effective ways to combat free radical damage. Colorful fruits and vegetables are excellent sources of these protective compounds. Different foods contain unique antioxidants that target various types of free radicals. Berries, dark leafy greens, nuts, and green tea are particularly potent sources. Each food group provides specific antioxidants that protect different parts of your cells. Vitamin C, vitamin E, and polyphenols are among the most important antioxidant nutrients. Exercise presents an interesting paradox in terms of free radical production. In the short term, intense exercise temporarily increases free radical production due to higher oxygen consumption and metabolic activity. However, long-term regular exercise actually strengthens your body's antioxidant defense systems, making you more resistant to oxidative stress over time. For optimal benefits, aim for at least 150 minutes of moderate activity each week, combining both cardiovascular and strength training. Proper hydration and recovery time are equally important. Several lifestyle factors can significantly increase free radical production in your body. Smoking is particularly damaging, generating over 10 to the 15 free radicals per puff. Excessive alcohol consumption, unprotected sun exposure, chronic stress, and environmental pollutants also contribute significantly to oxidative stress. Sun exposure, in particular, creates reactive oxygen species that can lead to skin damage, premature aging, and increased pigmentation. Let's explore practical recommendations to minimize free radical damage and promote overall health. For optimal antioxidant protection, aim for at least 5 servings of colorful fruits and vegetables daily. Choose whole foods over processed ones, as they contain higher levels of natural antioxidants and protective phytonutrients. To reduce free radical damage, avoid smoking completely and limit secondhand smoke exposure. If you drink alcohol, do so in moderation, and always use sun protection with an SPF of at least 30, especially during peak sun hours. Managing stress is crucial for reducing oxidative damage. Daily mindfulness meditation, adequate sleep, and physical activity all help reduce chronic stress. Some adaptogenic herbs like ashwagandha may also help your body resist stress-induced free radical production. Remember that balance is key when it comes to antioxidant protection. Consistent, moderate changes to both diet and lifestyle will provide better protection than temporary extreme measures. The various antioxidants in your diet work together synergistically for maximum effect. Recent research on free radicals and oxidative stress has advanced significantly. Scientists now use sophisticated biomarkers, advanced imaging, and even machine learning to better understand these complex processes. Researchers are developing targeted antioxidant therapies tailored for specific diseases. These therapies use nanotechnology delivery systems to ensure antioxidants reach exactly where they're needed, and personalized approaches based on individual oxidative stress profiles. Mitochondria are the primary source of free radicals in our cells. New research focuses on mitochondria-targeted antioxidants like MitoQ and SS31 peptides that concentrate exactly where free radicals are produced. These are especially promising for neurological and age-related diseases. An exciting new understanding is emerging about hormesis, the concept that low levels of oxidative stress may actually be beneficial. These mild stressors activate our natural defense systems, similar to how exercise and caloric restriction work. It's a biological example of what doesn't kill you makes you stronger. Current research has revealed oxidative stress as a major factor in reproductive health issues, particularly male infertility. As shown in this diagram, various factors contribute to oxidative stress in spermatozoa, affecting sperm quality and function. Targeted antioxidant therapies show promise in addressing these specific challenges. As research advances, scientists now embrace a more balanced view of free radicals. While excess can be harmful, some free radical production is normal and necessary. These molecules play critical roles in cell signaling pathways and are essential for proper immune function. The key to health isn't eliminating free radicals entirely, but maintaining the right balance between oxidants and antioxidants. The future of free radical research lies in finding the optimal balance for each individual and condition, recognizing that both too few and too many free radicals can be problematic.
If you enjoyed this video and would like to practice your understanding, go to MatEazy.com. There, you'll find this lesson outlined and divided, with a short quiz to see how well you really understood it. And if you're hungry for more, we've got piles of other topics for you to explore. Start learning easier. Visit MatEazy today.